My pleasure to come here to show you our story about the C4 rice. And yeah, I'm from IRRI in Philippines, and I would like to tell you about how we engineer the rice from a C3 plant to the C4 plant. The first, I will introduce the rice. I think most of you know about the rice, but also about what is the difference of this photosynthetics with the other C4 plant, and also our C4 rice project. Also, I would like to describe the problem we face by engineering the rice as a C4, and how we engineer it. We have two approach. One is the molecular engineering. Just we install the C4 biochemistry enzyme into the rice. But the other thing is we also want to discover some new genes involved into the C4 anatomy and the C4 function. This is the IRRI. We are in the Philippines. It's about 60 kilometers south of the Manila, and in the city of Los Baños is a university town. Rice is alive. Actually, you can see most of the rice production is in Asia, and then the rice is the most important crop. Actually, it's more than half of the human population is depending on the rice. It's a staple food for them. And then the 90% of the rice production is in this area, and where have the 70% of the world's most poor people living it. So many people depending on this rice, and many is poor. And the first in the rice, the green revolution in rice, actually is IR8. It's created in IRRI. This IR8 actually is crossed between one high plant pecta with this Dijaojian showed white rice. And then after the crossing, they create this IR8. It showed this plant is more like a nutrient using efficiency and high yield, and then not so easy to logging. And you can see now more modern the varieties. Actually, most of them is coming from this IR8. And last year, we just celebrated the 50 years of IR8. But now we actually face two more extreme climate change. For example, heat, salt, drought, and flood. And also the other challenge is the human population is growing so fast. And then now the traditional breeding process actually cannot compete the require of the rice production in the next 20 years. So for example, this red line is the best on the current breeding process for the rice, but we still have a gap to fitting the human growing. So now we are thinking about it's a time to have the next green revolution. In our C4 project, actually we're thinking about to increasing the photosynthetic rate probably is one of the solutions. Why is C4 photosynthetic in the rice? It's because the photosynthetic rate in the C4 plant actually is double as the C3 plant. And then the yield increasing also is about 50% more than C3 plant. It also have the higher water using efficiency, nitrogen using efficiency, and then the light using efficiency. And why the C4 plant have higher photosynthetic rate is because this is C3 models of the C3 photosynthetic. C3 photosynthetic actually is mainly in the one cell type, is Mesophyll Cell. And then inside have rubisco. Rubisco is 70% of the protein in the rice leaf. And then why this is main enzyme doing the photosynthetics. Why plant rice needs so much rubisco is because the photosynthetic rate or the CO2 fixation rate of the rubisco is so low. They need to compete with oxygen. And oxygen is doing, like if they fix the oxygen, it's photoaspiration activity. And that one is actually producing the product have toxic for the plant. So plant need to using the other pathway to digest this photoaspiration product, and it is the energy, the waste energy. But have the other type of plant, C4 plant, they actually separate the photosynthetic into the two cell type. One is Mesophyll cell, and one is bundle sheath cell. So plant actually first fix the carbon dioxide, the CO2, into the C4 product and transport into the bundle sheath cell, and then decarboxylase again, release the CO2, and then do the Kevin cycle. In these two cell type, actually the rubisco only have the CO2 around it and no oxygen. So the photosynthetic activity or efficiency is much higher. But you also can see, because of these two cell type of photosynthetics, the structure of the leaf also look different between C3 and C4 plant. In C3 plant, because the photosynthetic is mainly in the mesophyll cell, so between this is two veins, in between it have like 7 to 12 the mesophyll cell. But if you see in the C4 plant, they actually have like a 2 cell between the vein. So it means one, this is the bundle sheath cell around the vein, one bundle sheath cell accompanied with one mesophyll cell. And then the bundle sheath cell also full of the chloroplasts, because those chloroplasts is doing the Kevin cycles, have rubisco inside of it. But after we see the structure is different and also the function of it is different, so when we decide how to make the C4 rice, we have some challenge you need to face. First one is structure. It means you can see the bundle sheath cell size is different. They have more the organelle in the bundle sheath cell. And function also, we need to install this C4 cycle into the rice, and then reduce the photoaspiration, and then change the rubisco partitioning, and etc. And then after we understand the mechanism of the C4 function and how can we install into a rice, we're going to do the genetic modification. This is our C4 group at IRRI. We are about 40 people working. Some people doing agronomy, some is like a cloning or molecular biologists. And we also working with other like C4 consortium from 12 institutes in 8 countries. And Paul Crick is our group leaders. And this is our strategy to make the engineering 2-cell C4 photosynthetic pathway in the rice. So first we have the C3 rice, and then we're going to put the biochemical change, the biochemical gene inside, and then the gene involving to the anatomy changes. And we also need to find out some other genes is involving to the fine-tuning, and that time we can have a C4 rice. So first is molecular engineering. So this is the model of the C4 cycles we would like to install into a rice. It's NADP-ME type. So we already successfully installed these 5 biochemical genes into the rice. We also knocked down the GDCH, is a photoaspiration gene, and also rubisco in the mesophyll cell. And we also install the transporters into the rice. This model actually also will tell you where those genes we engineering, where they should be expression, because they have 2 cell types. So we want some gene just specific expression in the mesophyll cell, and some genes only expression in the bundle sheath cells. And then because we already individually transform those genes into rice, so now we are already crossing them together into the one plant. So our most advanced line actually have this 10 genes in the one plant. So you can see this is when we do the Western blot, is the protein. We can see those genes, one is accumulated, and also some like GDC and Rubisco is knocked down it. And this is the phenotype of these 10 gene stocking plant. They are slightly smaller than the wild type, but they still like a rice. And then, yeah. First thing, after we create those plants, we need to first to check if they still have the similar function as we expect in the rice. Our gene is mainly from the maize, so we like isolated the sequence, the genome sequence or CDS sequence from the rice, and we install into the from the maize, and we store into the rice, and we're using the cell-specific promoter. So, for example, here is the GM-PAXI enzymes, and then this enzyme in the maize, you can see here. I hope you can see well. The color of the green is the protein we are interested in. So here is the GM-PAXI, and then the red one is the chloroplast, and then the blue one is cell wall. So in this case, in this gene, we're using the specific antibody just detect this protein. And then in maize, you can see they are only expression in the cytosolic, in the mesophyll cell. And then so in the middle part here, the bundle sheet is no signal of this PAXI. But in the wild type rice, because wild type don't have this GM-PAXI, so you see have no green signal. But if in the PAXI overexpression line in the rice, you can see they have a similar pattern as like they are in their maize. They're only in the mesophyll cell, but in the bundle sheet is empty. So that is how we detect like if this cell-specific expression still in the rice after we transform them into the rice. The other one is because those of the gene is biochemical gene, so they have some functioning of like change the metabolite profile. So we do GCMS, HPLC to check. Another thing is enzyme activity assay to see if those plants have higher activity. So for example, this one is MDH, the other maize genes, we can see they have the similar activity rate as like they are in the maize and much higher than in the white type of rice. We also do the photosynthetic measurement to see if we install the C4 photosynthetic gene, if we can have a higher photosynthetic. The other way to verify this C4 pathway when we install into rice is we can do the isotope labeling experiment. It's because normally in the atmosphere, most of the CO2 is 12 carbon. And then now we are labeling with 13C CO2. And this is the CO2 labeling chambers we designed at IRRI. So when we are supply the CO2 with 13C, we can see where is this CO2 going. The carbon dioxide, they are going to C3 cycle or C4 cycle. So this is a result that we got. And then you can see, like this one is PGA. It's like this 13C. If they go to the PGA, how much they go to PGA? PGA is the product of the Kevin cycle. It's C3 cycles. So after the 10 seconds of labeling, you can see they have, and to 10 to 12 seconds, you can see they are the transgenic line and wild type. They have a similar of the 13C in the PGA. But when we look at this E4 product malate, you can see our transgenic line have a higher malate amount compared with the wild type. So it means those transgenic plants have the C4 cycles in functioning the C4 cycle. But after we calculate how much of this CO2 get into a C3 cycle and how much they go to C4 cycle, we find out only have 5% of carbon dioxide during the photosynthetic. It's moved through of the malate, so it means they are in the C4 cycle. So it means 5% of photosynthetic is using in the C4 cycle. Actually, it's very low because we want to increasing their C4 photosynthetic rate in the rice. But we still have other challenge. It's because this is like a CO2 concentration mechanism, but we don't have it now in those plants because we only put into the C4 biochemical enzyme layer, but we didn't change their structure because the structure is very important, like one cell of mesophyll, one cell of Bangalore, and then they can have the CO2 concentration mechanism. So the other thing is, but this one, to find out the gene involved into the Gang's anatomy, C4 leaf anatomy, is very, very difficult. In IRRI, we're using the genetic screening approach. The first thing we do is like C4 plant actually is, evolution after the C3 plant. So ideally, it's like actually C3 plant supposed to have all the enzymes for developing to the C4 plant. So we can do is one thing is mutate C4 plant to see like if we mutate some of the gene, can we move C4 plant to the C3? The other way is if we active some of the rice genes, can we becoming like enhance the evolution, like the C3 plant becoming like C4? So this is two strategy we're doing. This is like leaf anatomy. I show you the difference between the C3 and C4 plant. So in C3 plant, you have more mesophyll cell, and C4 plant only have 2 mesophyll in between. So the vein density actually is higher in the C4 plant. So in this area, we're doing is like we have mutated those C4 plant, and we do the vein density screen. So we have this portable microscope, and we put the leaf on top of it, and we screen it. If we can see some of the lines have like a higher vein density, and we identify this is a line we are interested in. So we do first is the sorghum. We do the screening for 3-4 years, and then we screen like 1.6 million plants. And then we find out one of the plants, actually 2 plants of them, they have more mesophyll cell compared with the white type of the sorghums. And when we check what's the gene in this mutant, I say have 2 mutants, but fortunately or unfortunately, these 2 mutants actually coming out is that they involve into the same genes. It's this steroid biosynthetic genes. So they just in the different locations. And then but now we still need to go to the next phases that how this gene evolve into the Gang's anatomy. And this one also already published by our colleagues. So if you're interested, you can go to see. The other way is to rice activation target line. As I say, we activate some of the rice genes and to see if they also change the Gang's anatomy of the rice. So we have like those tDNA insertion have this certified as enhancer. So if they are, because we know when we do a transgenic transformation, the gene is actually random inserted into the rice genome. So if they are in front of some genes or in the downstream of some gene, we can see, and they also can randomly mutate some of the gene. And then we do the same thing, we screen it. So this is actually at IRRI, our transgenic field. And because the number of the we handle is really big, so instead of using the screen house, we're using the field. And here we screen like 33,000 mutant lines. And then this is one of the lines we find out they have a shorter distance between the vein, so higher vein density. When we look at the number of the gene, actually they are about like reduce just 1 or 2 cell type, cell numbers. But the interesting is like they actually change the mesophyll cell lobing. So this line, the normal rice have a lot of lobing, but this line have the less lobing. It's more like C4 C4 plant actually. So this one, we also checking what is the gene it is, and we find it's in this location they have a mutation. So the other thing is another thing is we need to verify what's the function of these genes. And this is the anatomy screening. Actually, we have a collaborator. They are doing is using the transgenic, transcriptome analyze, and then to see like what's the difference between C3 and C4, and have some candidate genes. The other one's fine tuning, because we also consider maybe have some other genes we didn't, not like the anatomy or the biochemical, they have like a fine tuning function in the C4 plant. So we're doing is a low CO2 screen of Steria from loss of the C4 function. Steria actually is also one of the C4 plant, but it's much smaller and life cycle shorter. So now it's a new model of the C4 plant. We do the research, and then the sequencing also already been done. And this is our low CO2 chamber. So you can see we have about this 10 of this chamber. You can change the CO2 concentration inside. And after we setting the CO2 very low, around 30 ppm, or like ambient, like a normal CO2 concentration is about 400. And then, but if we put into the low, we can set in this chamber to about 30. After put into the chamber, we can do it with the fluorescence, like using this machine to screen the FV of FM. FV of FM is one of the photosynthetic activity measuring methods. It's chlorophyll fluorescence. And here is like how we screen it. Normally, the top one is C3, like a rice. If you put the compensation point, CO2 compensation point, it's like if we grow in the plant in a very low CO2 concentration, the rice die. But if you see the C4 plant like Steria, still very green, although it's smaller, but they still can survive because the C4 plant have this CO2 concentration mechanism, but C3 have no. We're using this characters to screen like C4 mutants, Steria mutants. So if some mutants, after we put into the CO2, low CO2 chamber, after 96 hours, actually some mutants dying. So it means those mutants lost their CO2 concentration mechanism, and they lost their C4 function. But after they die, how can we knowing what gene is involved? So we develop the other way. It's we don't put 96 hours, we put 48 hours. And then those plants, actually they are not dying, but they are photosynthetic, already responded. So when we do FE over FM, the chlorophyll fluorescence, this is a mutant. They already have a low FE of FN. And the Y type is still very green, so it's still very healthy. So using this technique, we will select 5 mutants, like homozygous lines, and then we checking their photosynthetic rate. For example, this one is Y type, and this is a mutant. Mutants have high compensation point, CO2 compensation point, and low photosynthetic. And their 13C isotope compensation also between C3 and C4 plant. Next thing is we need to know what is this possible mutants of this line. So they may be Rubisco exchange, the cell wall composition, or other functions. So this line actually recently we sent into the sequence, and our result is coming out soon about what actually is what mutant it is. In the 2015, the MIT technology review saying like the C4 rice project actually is one of the 10 broken through technique, but we still have a long way to go. And here just summarize like what outcome and the perspective. Like here our progress is like we successful engineer C4 biochemistry into a rice. We screen it for the C4 genes controlling the leaf anatomy. And then this research actually contribute from the wide consortium around the world. but we still don't understand is like this fine-tuning biochemistry and also the engineer, the leaf anatomy in the rice still not yet. And the other thing is like those big research actually is not only to create the C4 rice, we also hope like this research is value for the rice research and also understands evolution and C4 plant also. And here I would like to thank to our funding donor, BNGF, and also the IRRI and the UK aid, and also all the C4 rice consortium around the world. And also thank you for your listening. Thanks.